In this series of videos, we will investigate the structure of the urinary system as it relates to the histology of it. The presenter have no conflicts, and the session objectives are listed here as well as in your lecture notes. So the overview of the kidney structure is shown in this picture here, and you are advised to review that so that you're familiar with all the labels that are presented here. In overview, the kidney is encapsulated by a dense irregular connective tissue, which also have some myofibroblasts. Internal to that, there is the cortex, which is being pointed out here, and it contains the renal corpuscles, which we will see later, also referred to as Bowman's corpuscles, as well as convoluted tubules, collecting ducts, and other associated vascular structure. Internal to that, relating from this dotted line to this dotted line here, is the medulla, and that contains straight tubules, which are the collecting ducts and associated vascular structure, which are also referred to as vasa rectae. Internal to that, we have calyces as well as the renal pelvis, and this is where the urine, once it's filtered through the kidney, is collected in a funnel-like structure and then transconducted through the ureter to the bladder. The other feature to note here is what we refer to as the lobe of the kidney. That is just a one unit which has the pyramid in the middle, and there is some definition of that in your lecture notes. This is an overview histological structure, what you will be expected to see in the lab. Again, you can see the cortex running from this point here to the left, and then the medulla from the boundary of the cortex and medulla to the right here. So one part here is referred to as the pyramid, as we saw in the previous slide, and then here we have, which we will see later, referred to as the medullary ray, and this is where a aggregation of the collecting ducts are in the cortex. We'll come to see that later on. Looking at kidney at a functional level is composed of a number of functional units, which we refer to as nephrons. In this diagram here, they are simplified as individual lines and how they relate to the pyramids. And then on this picture here on the right, you can see a more detailed structure of them in a more functional manner. And we will look at each one of them separately, starting with the functional unit we refer to as the nephron. So nephron is a single unit which is by itself, does all the original filtration work. Each kidney has about 1 million of these, so together they act to filter a large volume of blood and filter it into urine. The parts of the nephron, you can see here on this diagram here, there are two nephrons here, one here and one to the right. The parts that we're going to be talking about are the Bowman's capsule, proximal convoluted tubules, loop of Henle, which has its descending and ascending loops, as well as the distal convoluted tubules. It's not so clear in this picture here, but we'll talk about that in details. And by the way, this is Mr. Bowman who described the Bowman's capsule. So looking at the Bowman's capsule itself, it is a rather complex structure. And again, you are advised to spend some time looking at this diagram here, because that would make life much easier when you are in the lab and looking at histological sections. The capsule itself is a hollow spherical structure outlined here, for example, and it is lined with simple squamous epithelium. It is indented by this glomerulus, which refers to these blood vessels which are coming in and then forming a very intricate network of blood vessels, referring to as the afferent, which is coming in, and the efferent, which is going out. Just for visual, I have filled in these spaces so you can visualize how the blood is in these glomerulus apparatus here. So blood comes in and blood goes out here, and all the function of the kidney takes place in this particular unit here. There is the parietal layer of the capsule, which is being highlighted at this point by this pointer, and then there's the visceral layer. And the visceral layer is made up of these very specialized epithelial cells called podocytes, which are being colored here in green. And what you would also notice is that they are actually forming these very, very intricate connections with the blood vessels underneath to form these very fine slits through which the blood is being filtered from the blood vessels into the urinary space, which is here. So the urinary space is being highlighted here in purple. So this is where the urine is first filtered from the capillaries of the glomerulus here through the membrane, which we will see in a minute, and then into the urinary space here. One thing to note here, and that would help you in histology sections, is that we refer to here these capsules having a vascular pole, which is up here at 12 o'clock, and that's where the blood vessels are coming and leaving the capsule, and the urinary pole. This is where the urine is being passed from the capsule into the tubules. We will see in a minute. Here it is, the histological view. So here's the vascular pole, and here's the urinary pole. And then this space that you're seeing here, the pale space, is the urinary space where the urine is being filtered from the glomerulus and then being channeled out into these tubules here. So one thing that we should really be focusing on is these cells, which are podocytes, which are present on the visceral side of the glomerulus, and they form these extensive branches, which are referred to as pedicles or processes, which completely cover the blood vessels of capillaries of the glomerula itself. And in doing so, they are forming these primary and secondary structures, and which allows a fine slits to be formed in between these pedicles, which acts like a holes through which the urine can come out of the blood and into the urinary space. And one other thing to remember is that these slits are being controlled by these proteins, and so they contribute how the protein gets passed into the urine. One other aspect of this structure of endothelial cells or the blood vessels and the podocytes is this glomerular basement membrane. So this is the membrane that is in between the two cell types. So in this picture here, you can see the podocytes on the outside, and there'll be podocytes on the bottom here as well. And then there's blood vessels here, or the capillaries here. And then in between is this basement membrane. And so it's basically attaching endothelial cells on one side and podocytes through the other. You can see these slits that podocyte pedicles are making by these arrows pointed out in this inset here. The glomerular basement membrane is very important in acting as a barrier to how much protein actually leaves the blood and enters the urine. The basement membrane itself is composed of different types of collagens, as well as other non-collagenous proteins such as laminin, fibronectin, and proteoglycans. The glycosaminoglycans in the glomerular basement membrane carry a negative charge, and so that actually blocks the movement of proteins which are 70 kD or larger. In pathology, you would probably hear about this, that integrity of this membrane is important, and loss of that integrity leads to molecules, proteins leaving the blood into the urine in an uncontrolled fashion. So proteinuria being one of those symptoms. Here is a diagrammatical view of what we just discussed. So here's the capillary with blood going through it, and then the pale blue color here is the glomerular basement membrane, and then podocytes on the outside with these pedicles forming the filtration slits here. So the urine being filtered from the blood through the membrane and into this urinary space. One other structure that we should think about while we are actually in the Bowman's capsule or glomerulus itself is the mesangial cells. And so these are found in the Bowman's capsules and also in the periphery of the juxtaglomerular cells or the JG apparatus. Just to make that clear to you, I'm going to bring back the slide that we saw before. So now these are the cells that we are talking about, the extraglomerular mesangial cells, as well as in here in the Bowman's capsules. And so this will give you some idea where these cells are before we proceed any further. So these cells do endocytose immunocomplexes as well as plasma proteins, and so they maintain this basic function, normal function of the glomerular basement membrane. Their numbers increase and they start to secrete growth factors as well as interleukins and prostaglandins when the glomerular inflammation or other disease states happen.